Today we are going to discuss about cell cycle and its regulation. I am Dr. Rajeev Basumatary, Assistant Professor in the DR College, Golaghat. So let us first understand what is a cell cycle. The cell cycle is the overall events of changes that take place in a cell between two consecutive cell divisions. That means the events that is taking place during cell division, that is prophase, metaphase, anaphase, telophase, and after cell division, where the cells grow in size, the DNA is duplicated, the enzymes and other proteins are formed in preparation for the next cell division. The cell cycle can be broadly divided into two phases, that is M phase or the mitotic phase, where the cell divides, and then the interphase. So interphase is the phase between two consecutive uh, mitotic phases, and then interphase is again divided into three subphases, that is Z1 phase or the first growth phase, then S phase or the synthetic phase, and then Z2 or the second growth phase. Okay, these are the phases of the cell cycle, and this is M phase or the mitotic phase, and this phase you can divide it into four stages of the cell division, that is prophase, metaphase, anaphase, and then telophase. So during these four phases the cell will divide, and after the M phase is complete, so next phase is known as the Z1 phase or the first growth phase, so where the growth of the cell division cell takes place. And after Z1 phase, the next phase is going to be S phase or the synthetic phase, and then the next phase is Z2 phase or the second growth phase. And these three phases, that means Z1, S and Z2, these three together they are known as interphase. That means the interphase can be divided into three subphases, Z1, S phase and Z2 phase. During the mitotic phase, as I already have told, that cells will divide, and the division will take place in four phases, that is prophase, metaphase, anaphase and telophase. And following the M phase or mitotic phase, there is Z1 phase. So Z1 phase is also known as the first growth phase, during which the cell grow in size and synthesize various enzymes and nutrients that are needed later on for the DNA replication and cell division. So Z1 phase, during the overall summary of the Z1 phase is that the cell will grow in size, since before Z1 phase, during mitotic phase, the one cell was reduced in half of its size due to the division of the cytoplasm into two cells. So that is why that reduced cell size has to grow first, so that it can again divide in the next M phase. So during Z1 phase, the cell grows up to a certain extent, and then it will also prepare for next cell division, and it will synthesize some proteins so that and some nutrients so that the cell get nutrition. Then after Z1 phase, next phase is DNA replication. So during which the DNA in a chromosome, it is replicated. That means in a single chromosome there is only one DNA strand is there, DNA double helical strand is there, one DNA double helix is there, and during S phase that DNA is replicated so that there are two chromatids in a chromosome. And after S phase, next is Z2 phase, during which again cell will grow in size, they will synthesize proteins, and those proteins especially, which are going to help them during the M phase. Two distinct events takes place during cell cycle. That is, one event is related with the cytoplasm, and the next event is related with the chromosome. So that is why they can be said as cytoplasm cycle and chromosome cycle. So actually the cell cycle is composed of a cycle of cytoplasm and then another cycle of chromosome. So what happens, for example, say during the start of this M phase, the size of the cell is, let's assume it's 100% size, and after the M phase is completed, that means when the cell is divided into two daughter cells, the size is reduced. Actually the cytoplasm is divided into two cells and the size remains 50% in both the cells. Okay, that means the cell is reduced to 50%. And what happens during this Z1 phase? The cell grow in size. During S phase there is no cell growth, but the chromosome is divided, chromosome is replicated, and during Z2 phase again the cell will grow in size and it will again attain the 100% size. So this is known as the cytoplasm cycle. Okay, now let us understand the chromosome cycle of cell cycle. So at the start of the M phase, the chromosomes are like this, means they have two chromatids, two sister chromatids. So during metaphase and anaphase, these sister chromatids are separated and they will be separated as single chromatid, and each chromatid will be divided in two different daughter cells, and this chromatid will go, and during S phase there is replication of DNA, and after DNA replication the chromosome will again attain this structure, having two sister chromatid structure, and then it will again get divided into single chromatid structure after the M phase is completed. And this division actually takes place during metaphase and anaphase. During metaphase the spindle fiber is formed on both the side of the chromosome, and then this spindle fiber will separate the two chromatids, leading to single chromatid chromosome, and this is known as the chromosome cycle of the cell cycle. This cycle of cell division or the cell cycle is regulated by the cell, and there are mechanisms which regulate this cell cycle, and regulation is very much important. So let us understand how this cycle is regulated. So cell cycle regulation is a process that controls the series of events by which a cell goes through the cell cycle. When the cell cycle regulation doesn't happen correctly, the cell may divide in an uncontrolled way, and diseases such as cancer can occur. So that is why cell cycle regulation is an important mechanism for cell. It is a mechanism to ensure that all the events of each stages of the cell cycle is completed successfully, and ensure that no cells divide unless every necessary event is completed. So this is also another important point to understand, that the cell cycle actually ensures that every event has to be completed before the cell is divided. The regulation of the cell cycle, it works by two systems and involvement of various molecules. And the first system is the system that ensures the proper completion of all the assigned activities in each phases of the cell cycle, which we know as the cell cycle checkpoints. The function of these checkpoints is to check that every activity that is assigned in each phase of the cell cycle is completed properly. Otherwise, this checkpoint is a mechanism that will stop the cell at that point when the activities which has to be done are not done by the phases of the cell cycle. The next system is a system of molecules and enzymes, which is known as cyclin and the cyclin dependent kinases. This system actually they help cyclin and cyclin dependent kinases. They help the cell to cross the checkpoint and to continue the cell cycle. So this part we are going to discuss in later on. So today with this cell cycle chapter we are going to discuss about the cell cycle checkpoints, and about cyclins and CDKs we will discuss later on.
Okay, let us now discuss cell cycle checkpoints in detail. The cell cycle checkpoints are also known as restriction points, and they are the control mechanism in the eukaryotic cell cycle which ensure its proper progression. Or in other words, the checkpoints are the mechanisms to check and ensure the proper completion of each events assigned for each phase of the cell cycle. So this system acts like a timer or a clock which sets a fixed amount of time for each cell to spend in each phase of the cell cycle, while at the same time it also responds to information received from the processes it controls. It also senses the cell cycle checkpoint. It can sense or detect the defects that occur during the DNA replication, and if the defects are detected, then the cell cycle is arrested in response until the defects are repaired.
There are three checkpoints or restriction points in the cell cycle. They are Z1 checkpoint, another is Z2m checkpoint, and then Metaphase and Anaphase checkpoint. So the Z1 checkpoint is located here. That is during the Z1 phase. This is called G1 checkpoint. And number one. Next checkpoint is between the Z2 phase and M phase. So here at this point, at this time, here is another checkpoint or restriction point which is known as Z2m, number 2. And the third checkpoint is known as Mitotic checkpoint or Metaphase Anaphase checkpoint, because it is located in this point. During the metaphase and in between the metaphase and the anaphase of the M phase. This is metaphase, anaphase checkpoint. Every checkpoint has some functions assigned to them, and the G1 checkpoint it checks for cell cycle, that is whether the cell has grown up to its mark or not, whether the cell has attained a considerable size after Z1 phase. Then next it will check for availability of nutrients. So nutrient availability is important for the cell if the cell has to proceed for next cell division. So availability of the nutrient or availability of favorable environment is checked at this checkpoint, that is G1 checkpoint. Next it will check whether there is any damage in DNA damage. So it will be checked in this checkpoint. And the next it will check for growth factors that are needed for the cell to grow. So these are the things which the Z1 checkpoint is going to check, that is cell size. Cell has attained a considered good size or not. It will check for availability of nutrients. It will check for damage in DNA. Then it will check for growth factors. If these conditions are met, then the cell will cross this checkpoint, Z1 checkpoint, and it will go to the S phase where the DNA replication will take place. If these conditions are not fulfilled, then the cell will not going to enter into the S phase, and it will be diverted to another stage which is known as Z0 phase. Okay, so it is like a stoppage where the cell will stop, and it will not go for cell division anymore. So if when these conditions are made, again these conditions are fulfilled, that means nutrients become available, then growth factors are available, then at the time the cell will again go from Z0 phase to S phase. The Z2m checkpoint, which is here in the middle of Z2 phase and M phase, the Z2 point checkpoint, it checks for cell size, whether the cell has attained a complete size that is eligible for division, and then next is DNA replication, whether all the DNA in all the chromosome have been replicated or not. If these two functions are not fulfilled, or the DNA replication is not complete, then the cell will be checked, or it will be stopped at this point, and it will not enter the M phase or the division phase. The third checkpoint is the Metaphase Anaphase checkpoint, or it is also known as Spindle Checkpoint, because the function of this checkpoint, which is located between the transition of metaphase and the anaphase in mitotic division, the function of this checkpoint is to check whether all the chromosomes have been attached by the spindle fiber on both sides. So if the spindle fiber is not attached to the chromosome, then there will be faulty separation of the chromatids during anaphase, and as a result one cell may not get a chromosome strand because there will be no separation of the chromatids if the spindle fiber is not attached from both sides. So that is why this checkpoint is one of the most important checkpoint, which ensures that all the spindle fiber is formed and it is attached with all the chromosomes. This is all about the cell cycle and checkpoints in the cell cycle. So in the next class we'll discuss on regulation, that means how the cyclin and the cyclin dependent kinases, they regulate the cell cycle, or they help in crossing these checkpoints. They help the cell in crossing these checkpoints. Thank you.